aren't here to do this live and in person. Okay. Gametes produced by an organism contain a combination of genes from that organism. In every gamete, this combination is, remember what a gamete is? A gamete is a sperm cell or an egg cell. Okay. And if we click on the link that I gave you, it'll show you, and I know the picture is a little bit blurry, I apologize, but it still shows you what it needs to show you. So during the process of meiosis, which is how we make gametes, which are sperm and egg cells, okay? So meiosis is what your body does when it's creating sperm cells and egg cells. And in the process of creating sperm cells and egg cells, this thing called crossing over happens, where the DNA basically shuffles. It's like, if you think of your DNA like a deck of cards, when your body creates sperm and egg cells, it shuffles the DNA like a deck of cards and mixes them all up, and then takes half of that deck and puts them into the sperm cell, or in the female, half of that deck and puts them into the egg cell. Okay, so what this does is it basically just makes the genes that the baby gets from the mom and dad random. So the question is, in every gamete, this combination is what? So the combination of genes in every sperm cell and egg cell, again, is a little bit different due to this thing called independent assortment, or crossing over, same thing, which is this. Okay? Where the chromosomes swap DNA and they shuffle to make each one a little bit different. Okay. All right. You've seen this question recently on the evolution section whenever we were talking about reproductive success. And so deer have this seasonal breeding cycle. I mentioned something about how deer season, if you've ever hunted, opens at the end of November, and the reason why that happens is because deer only breed, meaning they perform the act necessary to create a baby. That's not when they give birth. Breeding isn't when they give birth. Breeding is whenever they do the deed, right? So that breeding season is just for 3 short months, and you might think that it would be beneficial for an animal to just breed as much as they can whenever they can. However, because it takes a lot of time and energy to not only have the baby, but raise it, they want to make sure that if they do have the baby, that they have it when it actually has the best chance of survival. The best chance of survival for baby deer is in May, June, July. That's whenever they actually have the offspring. So if they want to have them during this time, they need to breed during this time in the fall. The question is, what is the most likely explanation for deer having this seasonal breeding cycle? Well, it's like I just told you, they want to make sure that if they're going to go through all the time and effort of having a baby, that they want that baby to survive. So they want it to be born at a time where it is most likely to survive, meaning when there's the most amount of food available and it's warm, so the baby's not going to freeze to death. So which answer choice says that? I'll let you tell me. We'll come back to it if you need to, okay? No, it's not G.
It's H.
It is H, correct. Yep, it is H. Giving birth only in the spring and summer ensures that offspring are born when food is most available. Okay, this one should be very easy for you. So all you have to do is remember GCAT. GCAT. GCAT not only tells you the 4 bases that DNA is made up of, but it also tells you what pairs with what. The first 2 letters, GC, pair together. The last 2 letters, AT, pair together. So if the question is, what is the complementary DNA strand for this segment, what would A pair with? What would G pair with? What would G pair with? You can just look at those first three and see what those first three pair with, and there's only one answer choice that starts with the correct first three bases. Correct. For the purposes of the video, I'm not going to say the answer. I'm just going to say you tell me, and then the people watching the video can tell me. So if you guys hear me not saying the answer choice, the letter, then that's why. I want the people watching this to have to think a little bit. Okay. Which list describes the types of relationships the sunfish has with the other marine organisms? Well, we got to go back and read. That is an ugly looking fish though, isn't it? Woo. The ocean sunfish, or mola mola, is a large flat fish that spends most of its time in deep water, feeding mainly on jellyfish. They often have many species of copepods, which are small crustaceans, that bury their heads into the soft tissue of the sunfish. Sunfish will swim to the surface and lay sideways and allow the seabirds to eat those copepods from their skin. Okay, so copepods to them are kind of like how fleas and ticks are to us. You know, like ticks will stick onto you and suck your blood, right? Copepods do the same thing to the sunfish. So the question is, what type of relationships do they have with each of those three other animals that they mentioned? So we have seabirds, jellyfish, and the copepods. Let's start with what we know. What is one type of relationship that we know between mutualism, parasitism, commensalism, predation, all of those different relationships that we know about? So what type of relationship do you think that the sunfish has with the jellyfish? They are eating the jellyfish. So what type of relationship is that? It's not one of the symbiotic relationships here. These are the three symbiotic relationships. Remember, happy, 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 meh, happy, sad. Okay, happy, happy is mutualism, happy, meh is commensalism, happy, sad is parasitism. All right. So, what type of relationship, though, do the jellyfish have with the sunfish? The sunfish are eating the jellyfish, so what would you call that? Think of a blank prey relationship. I'll let you go. Okay, let's try something else. All right, let's try, what do you think, how do you think the copepods interact with the sunfish? What type of relationship do you think that they have? Remember, the copepods are like ticks. They bite onto the sunfish and they feed off of the sunfish. What do you call that? Predation would actually be when one thing eats another. In this case, they are feeding off the sunfish, but they're not eating the sunfish like it's not a predator-prey relationship. It's like a tick on you. The tick is technically eating off of you, but it's not like eating your whole body, right? So would that be mutualism, commensalism, or parasitism? There's like literally, it shows it in the picture right here. There's the tick on the dog. Yeah, parasite, right? So that's parasitism. So how many answer choices say that there is a parasitic relationship with the copepods? Is that the only one?
Yeah.
Yes, it is, right? So the only one that says that they have a parasitic relationship with these copepods, well, you tell me, even though I just pointed to it. Yeah, like I said for the video, I'm gonna make, because what people do is they just fast forward through the video and then they just wait for me to say a letter and then they just put that, but they don't really invest any time thinking about it. So I'm gonna try not to do that. All right, which statement accurately describes the energy needs for photosynthesis and cell respiration? This one should be easy. So in other words, where do plants get their energy from to do photosynthesis? The sunlight, right? And where do we get our energy from to do cell respiration? We get it from food, right? So I put in here in the hints, chemical energy just means energy from food. And the word solar means sun. So solar energy means sun energy. Chemical energy means food energy. Okay. So you're going to tell me the answer choice that says that plants need solar energy to do photosynthesis, and chemical energy is needed for cell respiration. Okay, so you're going to tell me whatever one that is, and I'm going to scroll on, and I'm assuming that you guys are good on that. If not, we can always come back at the end, and if you need me to go back over, okay? All right, which table shows the 2 steps of DNA replication? As a friendly reminder, remember that DNA is GCat and RNA is GCal. So remember what RNA has that DNA doesn't have. If you see the letter U or uracil, U stands for uracil. If you see the word uracil, you know that they're not talking about DNA, they're talking about RNA. So which 2 can we rule out right away because we know that DNA is not being involved? In other words, which 2 can we rule out because they mention uracil in step 1 or U? G and J? There's no uracil in G, but you got J correct. F does. Oh, I'm sorry. No, it does. You're right. Yeah. I'm sorry. I missed that. Yes. So there's actually 3. There's 3 answer choices that we can rule out right away because it mentions uracil, which is the U, which is only found in RNA because RNA is GCAU, GCal, instead of GCAT, GCat. So which one does not have uracil or U in step 1, mentioned in step 1? You tell me. That is correct, but I'm going to let the people watching this video tell me. Okay. Which statement is supported by this cladogram? This is just a family tree. This just shows you how closely related things are. So let's take a look. The only way to do this is just to look at each answer choice and rule them out. A, hippopotamuses are more closely related to cows than to javelinas. Well, find the hippopotamus. Here it is. Are they more closely related to cows than javelinas? Yes, they are. That is true. So we can pretty much stop there. But just to prove it to you, let's pick one of these other ones, like toothed whales are more closely related to mouse deer than to hippopotamuses. If you go up here to toothed whale, it says they're more closely related to mouse deer than they are to hippopotamus, and that's not true, obviously. So, all right, that one was pretty easy. Some species of millipedes will roll into a ball when threatened, while other species of millipedes can secrete noxious chemicals from their bodies. These adaptations allow the millipedes to do what? Why would they roll into a ball when they're threatened or secrete a noxious chemical from their body? Seems like they're trying to keep things away or get away from something, right? So which answer choice do you think sounds like they're trying to not get killed? Correct. That one's pretty easy. My hint on that one was use common sense. That's all you got to do, okay? Has nothing to do with different temperatures or amounts of energy or blending into different types of environments, okay? I would hope that you guys could figure that one out on your own. Yes. Can I? Hey. Okay. Yeah, let me come back to it. Let me finish this video and I'll come back. All right, so which of the following best explains why cells that contain the same genetic material, or in other words, the same DNA, because that's what DNA is genetic material, right? Why, even though these cells have the same DNA, do they not develop in the same types of cells? So here's my reminder. Remember, all cells have all genes, but only turn on the genes that they need to become what they need to become. Hold on. That's not right. But they only turn on the ones that they need to become what they need to become. That's what I was trying to say. Okay. Yeah, I did type that right. I just wrote it really weird. All cells have all the DNA, but they only turn on the genes that they need to in order to become what they need to become. Okay. So if we look at this picture here, this stem cell here, this is a cell, this is like a baby cell, and a baby cell can grow up to be, all baby cells are the same. But at some point, these cells get triggered, like there's some sort of environmental trigger that activates certain genes in the stem cell that makes them become muscle cells. So if they turn on the muscle cell genes, they become a muscle cell. If they turn on fat cell genes, they become a fat cell. If bone cell genes get turned on, they become a bone cell. Same thing with all these other cell types, okay? So all baby cells start off the same, and they all have all the genes to become any type of cell that they want to become. But they get triggered and activated at some point, and depending on what genes get turned on, that's gene expression, right? Depending on what genes get turned on will determine what type of cell they grow up to be. So which of these basically says that? Says that cells can become any type of cell depending on what genes get turned on. I'm going to keep scrolling. We'll come back if you need to, okay? If you don't know that one, just keep it to the side. We'll come back to it afterward. Okay. Although these proteins have different functions, they all do what? So what are we looking at here? Do you guys know what this is? This double membrane here, this 2 layers of membranes here. This is the cell membrane, okay? This is the thing that surrounds the outside of the cell. And in the membrane are these big proteins, and they all do different things. This is how, these are transport proteins, right? This is how stuff gets into or out of the cell. So although these proteins all have different specific functions, they all do what? They all allow things into and out of the cell, or allow things from outside the cell to interact with it to make things happen inside the cell. So does that sound like they're stopping chemical reactions within the cell? These things are not stopping chemical reactions from happening. They're actually helping them to happen, because they're making these chemicals and these molecules go in and out or make things happen. Are they synthesizing molecules that signal other cells? No. Are they helping the cell interact with its external environment? Or are they removing large waste particles from the cytoplasm? Well, they're not all removing large waste particles. In fact, I don't see anything going, well, yeah, I do. They're leaving the cell there, but they're not all making things leave. Some are bringing stuff in. So, using process of elimination and common sense, you can probably tell which one that is. And if you don't know, we'll come back. Okay, this picture here, it says the results shown in the picture were obtained using a technique known as gel electrophoresis. You don't need to know what that means. All you need to know is that this technique, it separates mixtures of DNA and it will show you side by side how close the DNA is. So this is like you're looking at the DNA. All you need to know is that this is one person, this is another person, this is another person, this is another person. And you see these little bands, these bright bands that are on each strip? The closer that they match, the more closer related they are. Okay, so if this is person 1 and this is person 2, they're not even close. So these two are probably not related. Okay. So basically what we're doing is we're just looking at the DNA of these different people side by side and comparing them. And what does comparing DNA between different organisms tell you? If I take the DNA of 9 different people and I look at them all side by side, what am I probably trying to do? Correct. I'm trying to see how closely related they are. I want to see, is their DNA very similar or is their DNA very different? Okay, this one, the answer is in the question. Like it literally tells you the answer. If you just read the answer choice, it will match exactly what is happening in the picture. All you have to do is say what's happening in the picture in words. Which statement best explains the function of the vesicles? Well, what's happening? We have the Golgi apparatus. We have these vesicles coming off of it, and they're moving to the cell membrane, and stuff is going out to the outside. So which one of these does that sound like? F, G, H, or J? Well, you can rule out the ones that sound like nothing's happening. I mean, there's only 2 that it sounds like could even be possible. No. Correct. Yep. So you can rule out G and H because we're not exchanging genetic information. And remember, genetic information means DNA. DNA is only found in the nucleus. So if you don't see nucleus anywhere, then there's no involvement of DNA. Extracting portions of the Golgi to be regenerated for growth within the cell. There's nothing happening in the cell. These pieces of the Golgi are being taken out and released outside the cell. So there's nothing happening within the cell here. So it's not G or H, and I'll let you tell me which one it is. Okay, based on these data, this enzyme functions best at what temperature and pH? Okay, so we have enzyme reaction rates. We have how active the enzyme is. That's what this whole graph is about. And there's actually 2 graphs. There's temperature and there's pH. So here's the hint, find where the highest point is. So it's going to be most active at the highest point, right? Because that's the rate of reaction. Rate of reaction just means how fast is the reaction happening. If it's happening faster, then that means it's more active. So the highest point is going to be the highest rate of reaction or the highest amount of activity. And what temperature does the highest amount of activity appear to be happening at? And what pH does the highest amount of activity appear to be happening at? Correct. Well, on that one, all you have to do is find the highest point, follow it down, see what temperature and pH that is, and pick that answer. Okay. Okay. How does the appearance of these butterflies help them to survive? Well, if we go back and read, it says the Indian leaf butterfly has traits that allow it to resemble a leaf. That sounds like camouflage, right? Blending in. The bright colors of the monarch, however, they stand out. They don't help it blend in, but what they do is they indicate that the butterfly tastes bad and can be poisonous, which is true. I've heard they do taste bad. Well, I haven't eaten one, but how does the appearance of these butterflies help them to survive? Because there's two different strategies here. One is trying to blend in and one is trying to stand out. So you just have to tell me, and again, the answer is in the question. Like, they tell you what you need to know. You literally just have to put it into answer form. So the Indian leaf butterfly is trying to blend in and avoid predators, and the monarch butterfly is trying to stand out and warn them and scare them away. So you just tell me what answer choice says that. Okay, the female reproductive and endocrine systems work interactively for which main purpose? Okay, so this one should be incredibly easy because we know reproductive system. What are we talking about? We're talking about making babies, right? So there's only one answer choice that mentions making babies. You don't have to be an expert here. All you have to do is know that reproductive means reproducing, means making babies. There's only one answer choice that says that. You guys know what it is? Okay, good. All right. You might remember this question. This is something that we talked about recently in Unit 11 with evolution, when we were talking about comparing DNA. So this question gives you an amino acid sequence for 4 different organisms, organism 1, 2, 3, and 4. Remember, amino acids make proteins, but what has the instructions on which proteins and how to make them? DNA, right? So if I look at the proteins in my body and compare them to the proteins in your body, and if they're different, then that means our DNA is different, right? So different proteins means different DNA, because DNA is what has the instructions on which proteins to make and how to make them. So which organism is the least related to the other 3? In other words, which organism has the amino acids that make up the protein that are more different from the other 3? So we want the sequence to be the same. So let's look at the first one. In organism 1, we have none of them have the first amino acid in common. Let's look at the second one. 1, 3, and 4 have the second amino acid in common. The third amino acid, only 3 and 4 have in common, and the fourth amino acid, 1, 3, and 4 have in common. So it seems like 3 and 4 are very closely related. 1 is also closely related to 3 and 4, but which organism has none of the 4 in common? Right. Okay. How is this method effective in fighting viral diseases? What method? The weakened viruses could be injected into healthy individuals. These are just vaccines. Okay, so you guys, all you need to know is that a vaccine has a weakened version of the virus. They literally give you a weakened version of the flu when you get the flu shot or the COVID vaccine or whatever. They give you a weakened version of that virus so that your body can have like a practice run at killing the virus, so that if the real thing ever comes along, they will already know how to defeat it, and it'll defeat it faster. That's the idea. That doesn't always work, okay? You could get the vaccine, it's not immunity. You can get a vaccine and still get sick later from the thing. It's like saying just because you practiced playing this other team in basketball, like you have practice and you practice playing them, well, when you go and you play the actual game against the actual team, it doesn't mean you're going to win just because you practiced. So it's not a guarantee, but it does help. Okay? But it could also make you sick. The actual practice round could make you like real sick. So that's another thing. But anyway, how is this method effective in fighting viral diseases? Yes. So the immune system, what it does is it develops these things called antibodies, which are just things that your body uses to kill viruses. And I just gave you the answer because I clicked on it on accident. Okay. All right. An individual with AO genotype and an individual with BO genotype can produce offspring with which of the following genotypes? All right, so you guys remember Punnett squares? No. This is a Punnett square. What they're saying is if you have a mom that has AO, which is A here and O here, and then a dad that has BO, B here, O here, remember you're working like a times table? So you're going to have to use your imagination because I don't have one that says AO and BO. But if this is A and this is B, what would go here in this box? AB, right? If this is O and this is B, what would go here? BO, right? So the blood type would just be B because A and B are both dominant over O. So this would be AB, B. If this is A and this is O, what would go here? AO, which would make the blood type A. Or if this is O and this is O, what would go here? OO, which would make the blood type O. So we can have A, AB, B, or O. We can have all four blood types from these two parents. Which of these best demonstrates mutualism between certain types of bacteria and humans? Remember that mutualism is happy, happy. Remember, sorry, this picture's so blurry. I know it stinks, but that says mutualism. And you can see two happy faces. They're both happy, happy, happy. So which of these describes a situation in where both the bacteria and the humans are happy? Thank you. Well, D, we can rule out D because food poisoning doesn't sound like a happy thing for people. B says bacteria become resistant to medication that humans use for treatment. So that doesn't sound like it's good for people because it's So it's either A or C, but A says that bacteria at the site of injury produces toxins that damage healthy tissues. That does not sound like it's happy for the person, right? So process of elimination will give you the answer on that one. Just remember mutualism is happy, happy. But you might have a question about how commensalism or parasitism works. You just have to remember that commensalism is happy, meh. Parasitism is happy, sad. What is the role of mRNA in expressing specialized structures? Well, when they say specialized structures, what they're saying is proteins. Now, you may not know that, but all you have, if you just delete that second half of the question, it just says, what is the role of mRNA? That's the same question. So here's my hint. You have to remember that DNA gets rewritten as RNA, mRNA, and that mRNA is used to make a protein. So mRNA is just a copy of DNA. So why doesn't it just go straight from DNA to the protein? Why do we take DNA and then rewrite it and then make a protein? Why don't we just take DNA and use that to make the protein? Well, it's because the DNA is trapped inside the nucleus. The DNA cannot leave the nucleus. The proteins are made outside the nucleus, but the DNA is trapped inside the nucleus, and it cannot leave. So DNA has to pass on the message to mRNA, and mRNA can leave the nucleus and go outside the nucleus where the proteins are actually made. That's why DNA has to be rewritten into mRNA first, because it's trapped inside the nucleus. It cannot leave. So what is the role of mRNA? Well, it takes the instructions from DNA and carries those instructions to the place where the protein is made in the cell, and uses those, and helps the protein to be made. So you tell me which one that is. And that is the ball game. Doot, doot.